Gorgia. So diversity here is probably between uh, 15 and 20 species. If we were to sit down and count them all up. But even on dive like yesterday's at the summit, it was not impossible that we were getting in excess of maybe 30 species, which is very typical. As we get shallower, we have more species diversity usually.
Hercules and Atalanta gauges look good. Comps are good.
Roger that, thank you.
Heteropathes as well. And then we've got sponges too. We've got several sponges. We collected a very unusual morphology a bit earlier, possibly a demosponge. But we've been seeing a lot of these Pharea turbocharger sponges, as they're called informally.
Can you full screen that
Wait,
guy?
did you say turbocharger sponge?
So useless.
Yeah, that's the name of the morphology that's been given by folks who helped annotate and identify some of these Pharea. There's not a lot of species names assigned to Pharea, especially for some of the newer, more newly collected species. Very complex group, very speciose group, so it was named so by the characteristics of the morphology. But I couldn't tell you, honestly, if it resembles a turbocharger or not. That's what it's called.
All right, who knows? Like a car turbocharger? Is that what we're going for?
I don't know.
Is that like the one that sticks out of the hood kind of thing? Is that what a turbocharger is?
All of us are super into cars.
Clearly.
Is anybody really into cars? Dan?
I mean, zoom, but
What's that?
What is that
Sorry,
mean?
I was sending a screenshot to one of our software engineers. She's.
are you into cars, Dan?
Do you know what a turbocharger looks like? That's the
Yeah, I know what a turbocharger looks
Does
like.
this sponge look like a turbocharger? Follow-up question.
This one is polyopagon.
Say a
This
Nautilus
Oh,
looks
oh,
more like a
okay.
does not belong
turbocharger.
to that designation.
Sorry, never mind.
Doesn't look like the turbocharger on my truck. I can tell you that.
Well, that's good. It's not a turbocharger sponge.
I think maybe
It's
we
more
can
like
grab
a
a rock
soap
before we
dish.
get to the cliff.
Yeah, I think we can do a rock.
Okay.
How many open boxes do we have?
We have
Oh,
a lot of open
plenty,
boxes, yeah.
Plenty.
Yeah.
Yeah, let's do it.
Okay. I will keep my eyes open.
We have been faithfully taking rock samples every couple 100 meters along transect so far. We are
So
we need to pick up the pace a little bit
I'm fairly
after
stretched
the
out
rock
here.
collection, though.
Okay, I'll go quickly. Let me see.
But after we get across this,
Maybe this guy down here at the
slope
bottom
coming up.
left of the screen.
I'm going to try this one,
Okay.
Gibson, because there's a rumored issue with the manipulator. You see any hair you like, or should
Yep. Copy that.
This guy maybe?
I? Right up. I want to test our manip and see if the girl said it had some issues.
Yeah, it sounded like it was running away from them. Speaking of rocks, someone in the chat wants to know, what do the rock samples actually teach you?
Can you hit 5 on the gizmo?
This is a total softball question for Brandon. Teed up
Thank you for
to
the chat.
crush it.
Actually, I can
So they can teach us multiple things, but initial observations once we open the rock is what kind of processes are going down on this seamount. So if you open the rock and we see this nice gray, what we call basalt, then alteration hasn't penetrated the rock yet.
Not be possible.
We can bring it to the lab and potentially date it. Otherwise, you can see yellow or reddish alterations on them, and
Well, it's 'cause there's no
that ruins the elemental compositions and can't date it anymore, but
telemetry.
gives us a little insight.
See that manipulator try to punch me in the eye?
I did.
Yikes.
You could also, yeah, there's some of the stuff looks potentially breakable too. I'm surprised from these seamounts how easy it is to get rocks
Yeah, a lot of broken debris everywhere.
broken, and it's not one sheet of concrete crust.
That is not what I was expecting.
Yeah,
We've kind of been okay on getting samples off of it.
Yeah, we've been okay getting samples, but a lot of them have been bad.
Oh, okay.
So, you know.
I take it back.
So here's a question for Delta Dan. How far away are you from what's actually being filmed with the ROV?
Sorry, I didn't hear there. There was multiple conversations going on.
Would you like me? Are you ready to hear it?
What's that?
Are you ready to hear it?
Yeah.
How far away for you? How far away are you from what's being filmed from the ROV? Do the coral and fish have color, or are they also too far away?
Well, for example, that rock in the background is 5 meters according to the sonar.
Oh.
geologists get super bonus points for getting corals attached to rocks. Like many watch bucks?
Watch bugs.
Watch bugs.
Bringing it back.
Typically within a couple meters of what we're looking at. If we're picking something up, the maximum reach on the manipulator's 2 meters approximately. So we're usually within a meter of, so the lasers the lasers right now are probably 2 meters from the front of the vehicle.
So you need a spot to like sit down, is what you're saying?
What's that?
Do you need a spot to sit down then when I pick a rock? Is that what you mean?
Oh.
Or you're just
No,
talking
you
about
can
the
pick a rock anywhere on the screen and
okay.
I can get to it.
Sounds good.
Question there. I'm trying to get back in front of the Atalanta there. I was getting a tug.
Okay.
And
Yeah, that's nice.
Gibson, how far would you estimate the light pool is, or how big is the light pool? You know, if you're watching on channel 2 and you're looking at the Atalanta feed.
Oh, Hercules is probably a couple meters tall by 2.5, 3 meters long. So it's greater than the size of Hercules. It'd give it like, I don't know, 4 square meters probably. What do you think, Dan?
something like that.
Sorry, I'm in metric.
No, that's great.
We are a science vessel.
You lay a
We do
couple.
everything in metric except wrenches and speed.
What do you think
We're
about the
still
rock
not,
this coral's on right here?
You laid a few people end to end.
Yeah, I love
2
it.
people
two
in a
It looks
square.
nice and thick.
Four
Look back at
people in a
the
square.
some of the records there. Was it you with us, Steve? We got the big rock with the big coral on it.
I remember straining my back lifting a rock. Is that what you're talking about? 90
That's a
pounds.
sponge, though. That's a sponge on that one. Is that what you're talking about?
Yeah, this one right here.
And what type of sponge is that? It doesn't look like a glass sponge.
It's a dead one.
Well, okay.
I can
It might
tell
be
you
small
that.
actually, the rock.
That's,
But-
yeah, I mean,
Yeah.
the sponge is dead. It's no use for us
Okay.
biologically if there's a better rock.
No, yeah, let's not do it. It's smaller than I thought.
I'm gonna.
It's perfect for me to perfect my sponge necropsy technique. Figure out why it died. How old it
CSI
is.
sponge?
Yes.
Are we doing sponge forensics right now?
Lynette is, which is a new passion we have to hear all about.
Okay, I've got some problems with this manipulator here. This could be.
Speaking of sponges, someone in the chat asks, have any of you actually felt a live sponge?
Yeah,
Um,
Kukui,
Can
you
you
got
put your
to
finger
process
on
the
the
samples,
craft
right?
valve?
Kukui is
Your
indisposed.
mouse
Oh,
finger
hey,
on the
Taylor's
craft
here.
valve.
Yeah, filling in for Kukui. But
Sorry.
yes, we have touched
So
a
just
lot of
look at if
sponges
things go
recently,
hairy.
actually.
Yeah, if I
They all have
scream
different
like
textures.
a kindergartner.
You got it.
All
I
right.
don't know.
What are some of the textures like?
Some of them are actually pretty brittle and firm and not squishy like you would think.
And then obviously like the glass sponges, you might not want to squish. But then, yeah, some of them are very squishy and feel very similar to like a kitchen sponge. Yeah, but I guess they range in all different types of textures.
That one is
Do the glass sponges poke you?
Yeah, depending on how large their like spicules
Yeah, it
are.
looks nice and blocky.
Okay.
Do you want to see if you
It's
can
good
grab
to
that
know.
one as you're testing?
Here's actually a question for Steve. Steve, are you ready for this question?
ready.
Hang on, I just lost it.
You all right?
Does the depth
Yeah,
and available food
it
sources preclude predators
When
such
it
as
loses
sharks?
telemetry, it
No, there's definitely sharks here.
goes right spastic.
I don't know if
which is what
we see
happened
them
last
from time to time.
In the log
Or
that
actually,
it
there's a
did
coral
that.
on that one.
I might
too,
start
so
tugging on you soon here.
bonus sample.
Stay tuned, folks at Nautilus Live, because you may just see a shark.
So keep your finger on that button.
Finger is on the
I
button.
am getting tugged. You can zoom in there.
Actually, it might be a bryozoan.
What is a bryozoan?
Could I look at the bottom when you get a chance? The rock, or I guess the left side.
Let's get, well, we can zoom to confirm first before I put my foot in my mouth. I think it is a bryozoan, though. It's a
You can zoom
small
right in if you
invertebrate.
want. Well, maybe not quite that far.
There was a
They wanna
bunch
see
of
that
those
great.
bryozoans on that
now,
sponge
now you
as
can
well.
zoom in.
It's a very nice bryozoan.
Okay, what box is this going in? We're getting drug across the seabed here.
I think
We've
he wants
got
to
the
see.
starboard box open. If you think it'll fit in one of the smaller ones.
Yeah, it should, yeah.
Okay, I'm taking my finger off that button.
There's a black coral, too.
Right.
This is a black coral.
You all right with that?
Is it juvenile?
Very, very small one, yeah. I don't even know if you could
You can go wide.
tell much about it, but it is, yeah.
Do I
You
get
might
some
want
watch
to
bugs?
spin on that.
Yeah,
See if he
yeah, you
wants
get
it.
some watch bucks.
Oh, you helped me too, so.
You can redeem them for
Open the box.
nothing.
You want me to open the box?
You don't know they're
Aggressive.
redeemable for nothing. But we'll only know at the end.
No, that's not true.
After
You-
everyone banks
it's
all
not,
their
there's
watch
not
bucks.
nothing. You can redeem them for
Pizza.
ice cream bars.
What boxes are
Oh,
open
that's
here?
true. It's Sunday.
You can put it in bio box B, C, or D.
B, C, or D? We'll go for B.
Really quickly going to answer that question in chat. Why are you still using Atlanta? Argus has gone through some technical difficulties, and we are using Atlanta instead, but Argus will probably be back up by 2023.
Yeah, set a sample 072.
So bryozoan, black coral. Yeah, you got it. Awesome. They're probably representative of what's in this area, which is useful. Bryozoans are really poorly sampled because you could tell their size and they're very fragile. So any
Got the ground faults on
any
that.
bonus
DC.
collections really does help our understanding of biodiversity in this
relay
area.
isolation
Relay isolation. Yep.
and turn the DC bender and the DC ground fault sense back on.
Coming on.
Look at the ROV
No, there's no
oscillate
pressure,
when I do that.
but it's calculated from depth. Yeah.
All right, they're on.
And pressure would be in bars or millibars, I think, on the CTD.
So to answer that question in chat, not sure what depth, what pressure we're currently at.
Divide by 10, you get bars, or convert to feet and divide by 2 and you get PSI.
Or you go to atmospheres. Every 10 meters an atmosphere. So we're at
Much easier to
100
remember.
meters,
atmospheres.
190 atmospheres, and it's pretty close to a bar.
Tell us more, Gibson. How much pressure is in an atmosphere?
Atmosphere.
Like 190 atmosphere, I guess.
14 and change.
Atmosphere, there in one atmosphere there's 14 point something psi. So 14.7.
We got
Do 15 times
2K out of
190.
that exercise.
Nice.
Can you hit one on the?
Coming over.
Yeah, we're just a little bit under, or just
Just a
around
little bit.
a 90 standard piece of pressure.
Yeah, 190 bar, which you multiply 190 by 14.7, and that will give you the PSI, which is a lot. Folks at home, do the math. Come back to us.
All right. Bonus points, bonus watch bucks.
Yeah.
How many millimeters of mercury?
Yes. We will award watch bucks to viewers with us online.
You can cash them in at your local, I don't know.
I don't know. See, you gotta stay
interesting.
tuned to the whole expedition to find out what watch bucks can convert to. You gotta stick with us.
The net worth of watch bucks.
Yeah, it's gonna be 3 weeks of watch bucks.
Apparently the other two,
Still can't get all the way around to you here.
the
Really?
other two
Yeah.
dives have nicknames. So are we going to be watch bucks?
We
I
could
like
we could
watch
work.
bugs.
Yeah, we can work on it. They have the names. I feel like have to come to us.
Almost.
So that's a good one.
I
It's a
think
good candidate.
it's sea
There's
pickle
a big
or
rock in front
muted,
here.
and
Yeah,
now
there.
I think it's going to be watch bugs.
You're gonna have to come up a little bit, or you're gonna hit that big rock.
So ROV Argus
Altitude
thrusters
20.
are currently
go.
not functional, and that, as
Oh,
we
I
say,
went the
it's
wrong
knocked
way.
out for,
Yeah, I'm getting
or will
tugged.
be coming back in 2023. This is the last ROV expedition of the
They're gonna be-
2022 season. We have 5 more expeditions yet to come, but they won't be diving with Atalanta or
That's why.
Argus.
Turn this
So
up, turn
it
this
sounds,
down.
it's like that see you next year joke, right, of December. It sounds like a really far away, but it actually is just one of the tasks of the off-season maintenance period that's coming up.
I
Some
am not in a good
projects and
position.
overhauls on the ROVs, which will be really exciting.
That's a close
come
Well, there's
rock.
a fairly heavy sediment drape right here.
Sorry,
It's
I'm going to
kinda
tug you
different
again.
from what we were seeing
There we
earlier.
go.
Yeah, come up a
Interesting.
little faster.
Really
I
quickly
cannot
answering
make
that
this
question in
ROV
chat.
go where I want it.
Do fish or other animals ever bother your bots during these dives? Not really. Sometimes they may bump into them every so often. We've had a cutthroat eel kind of eye us
Okay.
up. But other than that, it's been pretty
Man, that's quite a
pretty
ledge.
peaceful.
The shrimp,
I managed
The
to
shrimp-
turn in
the
my
shrimp.
tether
they
too.
don't care.
Sorry.
Fearless. We have a honey badger of the ocean.
These
You're looking
these
down.
look
so
really
you probably are.
blocky
They do.
for basalt. Well, I guess you can have blocky basalts. No, is that common?
Yeah, I don't
Yeah.
see why it wouldn't be.
Ooh, look at this big coral on the other side.
Wow.
You're
Hello.
gonna have to.
Yeah.
Wow.
What type of coral can get? Oh, and that
So
sponge, too.
cool.
Yeah.
we're-
Actually,
we're
can you
not
drift
moving,
down?
so you're okay.
Drift down just off screen on the bottom. Something weird down there.
Our viewers at Nautilus Live, if you want to
Yeah,
go
coming
over to
out.
channel 2, you can see the entirety of that little
Can
sponge
tilt
area as
down
well.
a little more.
Yeah,
for me.
coming down.
No, just tilt
Tilting down.
All right.
Can we look here? Is that like a shadow
Interesting.
from an old holdfast or
Go ahead and zoom in there a little, Dan.
dark patch, circular? Yeah, that's what it looks like. Okay, just making sure.
So there have been big corals here for a
You
long
can
time.
see another one there too, yeah. It's like the 5 o'clock shadow.
It's like polyps or sea anemone marks.
Yeah, it could be from the anemone. Yeah, it could be
Okay,
from
go
a detached. There's a bunch of detached holdfasts down at the base of this incline, the slope.
So we have a preview in Atalanta there. It's a pretty spectacular clip
Yeah.
we're looking at here.
Yeah.
I think the current comes from this way.
It's really beautiful.
Yeah.
It's just covered with sponges. You can see all the white spots
Yeah,
in
these are
Atalanta there.
bamboo corals. They look like nodal branchers.
That's gorgeous.
That big fan there. Bunch of polyopagon satellite dishes. Another
Mhm.
common name. Great reception down here. We got Rhodaniridogorgia, a new species, that very small colony, the big one we saw yesterday.
Oh, where do you see that one?
Right under this sponge there.
Oh, okay, yeah.
Someplace along this wall, I think we'll
good
do
altitude
another
there.
Niskin bottle as well.
We'll
Yeah.
get some good
Do we have any sense of what the growth rate
Atlantic
of these
video,
sponges
Yeah.
is? Can we guess anything about their age?
I don't believe so. Not easily. Not. I don't think, or I'm not aware of any papers that have published polyopagon growth rates or ages. Can we zoom in on these? I think they're chrysigorgids here. High density cluster.
right.
They're incredible.
Go ahead, Dan. Zoom in from here. I'll try and dial my Z bias in and get a floaty.
Yeah. So this is chrysigorgia. Several high density colonies. So these are kind of planar or biplanar chrysigorgia. So that narrows it down to a few different species, given their size. Could be either very large Chrysigorgia chryseus colonies, or it could be Chrysigorgia scintillans, which is also known from around Hawaii, but also has a very wide distribution. I believe something like this has been sampled before, but can we stop and get a Niskin right here?
Yeah, I'd like to just look at the rest of this little cliff right here real quick, and then
Yep.
we'll come up above it and get a Niskin. Okay, go in.
While we're looking at this,
This is scintillans.
Delta Dan, I have a question for you from the chat.
Just want to see the top here,
What's
Steve.
what's flying the ROV like? Is it like a drone or some more of a land vehicle?
It's a fish as well, I
Combination.
think, maybe?
It's
Yeah,
there's
that's a halosaur. Almost certainly it's a halosaur.
Cool.
it's more like flying a forklift. Bread truck.
So fun.
Yeah.
And we have actually some questions for
Yeah,
Brandon
look
here.
at this.
Are basalt rock samples collected on ROV dice similar to basalt rocks found in Washington, Oregon, and Idaho?
Yeah, they're similar. Yeah, basalt is basalt. There's not too many differences, I think. The chemistry might be slightly different. Like,
It
I
appears
expect
to
to
go on
find.
for quite a while, Steve, as you can see now that
Yeah.
I'm lighting it up. So
You know what, I have some additional guidance.
I don't know where you want your Niskin.
Those that high density chrysigorgia, it doesn't have to be that cluster exactly, but I would like to grab a piece of that just to confirm its ID, because we have some records from Johnson Atoll and it's labeled as chrysigorgia planar, which is usually a good indication that it hasn't been collected and it's only been known by morphology from video.
Right there.
So Niskin and snip of that coral.
I would expect these to be tholeidic basalts, which is basically your generic basalt. It doesn't have abnormal concentrations
that,
in certain elements.
Is that one on the very top?
But I'm actually
Yeah,
not
the,
familiar
same
with
stuff, or-
everything here
West Coast.
is that same
So
species.
maybe
Yeah.
they can write it again
Right there.
and tell me about them.
This is a really nice one, too.
It is.
So as a little bit of a follow-up question, while we're collecting these basalts, someone in the chat wanted to know, do you ever find any prehistoric fossils inside of them?
No, so these are volcanic rocks, and magma will destroy life.
And then, I think I could actually answer this question in chat. Are white sponges dead, or is it just coral that turn white when they die? The white sponges are actually quite alive, and I believe most of these are glass sponge, and that is their natural sort of color. Correct me if I'm wrong, Steve, but I believe that's correct.
And they're also,
Can you say that again?
down here in the deep sea, corals come in a variety of colors, including white. That is their healthy coloration. So from shallow water corals, it sounds like this viewer is an ocean fan, knows about coral bleaching, which is one of the impacts of warmer oceans. And shallow water corals have symbionts called zooxanthellae, which are tiny plants and microbes that live within their tissues. It's a separate animal living within another animal symbiotically, and zooxanthellae are a big part of what give corals their color. And then when they get stressed out, they kick those roommates out. They kick out their zooxanthellae roommates. And that's what coral bleaching is that what you're left with looking behind is the clear or the uncolored tissue. So a lot of folks when they come down to the deep sea get surprised and concerned seeing corals that are white or lightly colored.
The still camp there.
And
Yeah, I'm trying to
instead,
park here so you'll
because
have
there's
time
no
to play with it.
sunlight, you don't need tiny animals that help make food and harvest sunlight
Choking.
inside your tissues. So the pigments that they create
Drop
are
the ISO a little bit and try
Okay.
again.
maybe
I think
secondary
it was just
metabolites or
let me
maybe
get
a
it,
little overexposed.
Let me get the ROV to stop and you'll get a better
Yeah,
Yeah,
collection's priority first.
let's just enjoy this beautiful view. We'll definitely get to more questions as soon as we can.
The
But
problem is I
absolutely
can
incredible.
actually see it, and which makes it, if I can't see it, I can usually nail it, but when I'm watching, I choke.
So what you want?
If you
Yeah,
want,
that's good.
while we're sitting here, you can square up Atalanta and play around with the
Yeah, that's
minis.
a
Use
really nice-
zoom. You might be able to get a good still for glory shot there. You can turn your head a little for a minute. Okay. I'm still a little bit leery of our nippinator here, so I'm gonna.
Are you gonna plan on going for the colony right in front of us?
I'm going to try
Turn
and
your
get the
toes
tether
there?
out of there.
Yeah, no
Um,
worries.
Are you happy with that one
There
right
we
in front
go.
of us?
Yeah, either, or the one off to the right that's in the lake pool, whatever you prefer.
Yeah, I gotcha.
It's all the same.
Very nice.
There we go.
There is a, if you grab this piece, there's a bonus anemone on there too. Alternatively, you can do something like that too.
Oh,
It's
hold
fine.
on, I'm not looking. I'm pushing buttons here. What am I doing? Can you hit 5 on the? Sorry, Steve. Say that again.
Yep, so you have a couple options. You can either, if you want a closer piece, you
zoom.
can do something like a cut there, or you can do a cut here for a bonus anemone.
Yeah, I can see if we can reach the bonus anemone. Before we do that, Dan, you want to do a full zoom there for me?
Sure thing.
So some polyps are closed and some are open, is that right?
Yeah, you can see the difference here.
While you guys are looking at the coral here, I'm just going to test the minute again.
Why do they open and close?
It could be many things. They could have just consumed a meal. They could have been, I don't know, brushed by some sort of animal like a shrimp or a brittle star that might cause local polyp retraction.
Okay.
Yeah, it could be many things.
Can you tilt up with the bubble
Thanks.
a little?
Back in the old days,
Chase me around there.
of
Sorry.
non-LED lights, getting up next to a coral for a very long period of time, the heat from the lights actually would cause them to
Okay, you can
respond.
tilt back down a little.
Interesting.
Will do.
I don't know what kind of lights they were. Were they
Okay,
like
pan
halogen
left a little.
lights in the old days?
Keep panning left, keep panning left. Yeah, they were halogens, and then we had CIDs. Is that right? Yeah, CIDs.
All right, ready to go when you are up front.
Pan left a little bit more.
While we're waiting, going to answer this question real quick. Are the ocean salts similar in comparison to basalts
bring
of
it back
the
up.
Northwest Pacific? Yes, they are.
Say again, Dan?
Tilt
They
down.
are pretty similar, although Brendan said that their mineral composition may be a little bit
down
different.
Yeah, they're different sources.
A little more.
More down?
Yeah. Down, down,
So
down.
the ones in
What a
Oregon
great view.
came from
How's that?
the Yellowstone bathypathes. Bathypathes just means
Okay.
large magma chamber.
Can you go wide, Dan?
Whereas the ones here came from mantle plumes. So I expect different composition, but still basalt. The major element chemistry will be the same. Yeah.
Zoom in there for me.
Yep. Oh, it's a crinoid, not an anemone.
You want the crinoid or?
Yeah, that's fine. Yep. This one can go in the forward bio box because I think it's going to come out in fragments, and it's probably a little more floaty than typical.
Right.
How did crinoids get their name?
Is there anything else floaty in the forward bio box?
I don't think there's anything in the forward box right now.
No, there's nothing in the forward box.
An incredible view, just layers and layers of chrysigorgia with the polyopagon in the back.
Yeah, wow. Those are huge sponges too.
Absolutely massive. It's a great view on channel 2 if you haven't checked that one out.
This one should cut or twist very easily.
Right.
Amazing.
Is that enough there? I can see if I can get on the
That's
branch.
enough, yep.
No.
That's all right. You can keep going for the coral.
Lost the critter there. I struggled on that one.
It was
That's like a
delicately
calendar.
done.
It feels like a page out of a
Do
calendar
a
right
full
there.
zoom there while we go.
Sure.
Hand, brand, logo, polyopagon.
Yeah, great.
Can go back to 5 on the bubble cam. Okay, you can go wide now, please.
So cool.
We're going to go in the forward box there?
That's the room, right?
All right.
We want to be careful when we should be okay porching out there. I don't think the DSC is going to hit anything, but keep an eye on it.
Will do. Let me know when you're ready.
Either side, Steve, or?
Yeah, dealer choice, yeah.
Roger. Okay, can porch out a bit there. That should be good.
Probably make a note, no rocks, wherever it goes.
Open a bit more for me. More.
What we usually did to pretty good effect in the past was keep one of the forward box partitions, for like bio only, or soft stuff that's floaty. We can double, triple, quadruple, whatever we need
Got
up.
a bumper there.
I think our tether's going to get... we got some slack still.
Speaking of these samples, we have a question for Taylor Ann, actually.
You want more?
How long does it take for you to process these samples once they've been collected? Which kind of samples take the longest? Which are the fastest?
It depends on, I guess, just how many samples we collect, but I'd say
Very nice.
if it's a really large, delicate thing, it could take a while if it has a lot of associates on it, and because we have to process each of those
Get
individually.
my snaggle teeth out of here.
But I feel like lately
Okay,
we've
you
been
can close
processing
it.
samples within like 2.5
Thank you.
hours. But depending on if we have push cores and different things like that, it
Well
can take a
done.
lot longer.
Why is that?
I feel like there are just different methods. Sometimes with push cores, you have to scrape off different
Zoom in one
layers
more
and
time,
put
yeah.
them in separate
Close it.
bags so that you can see what's in each layer of sediment, and you're not mixing them up. That's really important. Yeah, and that can take a while, and it gets really messy. that was sample number
Push some buttons
073.
This is painful. I got to turn our ground fault detect system off, then turn the manipulator on, then wait for comms, then do all the opposite.
We're going to do a Niskin pull here if you...
You want to do it right here or up above?
but
thanks
whatever's
for
more
reminding me.
convenient, yeah. We don't want to be too far above, no more than a meter or two off bottom or so.
It looks like the current's right in our...
Or just off a wall, it's fine too.
Yeah, current's coming to us, but that might be the thrusters pulling it in. I'll come up above the... Okay, you can go wide for me. See, we have Niskin number 1 down. That's...
Yeah, there's only been one pulled. I think it's number 1.
Go to hit button 5 for us, please.
Yep. Only number 1 has been pulled so far.
Go up, so we're actually over the colony here.
Nav, while we're doing this, can you drop a target here and call it like Coral Knoll or Coral and Sponge Knoll?
Coral Knoll, I like that. Coral Peak,
Okay.
Coral Knoll.
Like I said, we're not creative, but...
It's like coral connectivity knoll, like lots of satellites.
Could be Coral Wall, too.
Dan, you're a climber. What would you name this cliff?
Oh gosh, I'm more of a boulderer
Okay.
than like a tall wall climber. I wouldn't know. A fun looking one for sure.
And sometimes
it's got
that's all
extra
that
challenges,
matters.
like we gotta go around the sponges.
Look at that view.
Isn't it gorgeous?
And that was sample number 074.
I feel like I
Thank
want
you.
some of these like in my yard. Like they feel like sculptures to me. Like what if you could just have a forest of these like chilling at your house?
Yeah.
That would be pretty weird.
Someone says Coral Knoll feels like a retirement home. I kind of agree with you. Thanks. I could hang out here though for a long time. So
Yeah. Well, if I could live at this depth comfortably, absolutely.
yeah,
Here's one.
I need
Uh,
snacks.
A chat. What are Nitskin bottles and how do they work?
Taylor.
Oh, sorry, I was taking notes.
No
A
worries.
Niskin bottle is a bottle that we use to collect water samples at a certain depth. It's opened at 2 different ends. It's held open by bungee, kind of like rubber, almost like big rubber bands. And what you were seeing earlier was the pilot pulling these little levers with the arm with tape on them. And that was pulling the Niskin shut in that area of the water column, so it holds all of that water just from that area, so that we can get a glimpse of potentially what else is living in this area if we can't physically see it, what other corals, by using eDNA processing. That's some research that Dr. Meredith Everett is working on. So yeah, hopefully we're going to get some good signals and see what species are here in this area.
I'm just overwhelmed here, Steve, so if you see anything,
Yeah,
you
no, I'm
wanna
keeping
zoom
an eye
in
out.
on,
There's
let us know.
spend all day here.
What's that?
We could spend all day here, yeah.
Yeah, good.
Incredible.
2 meter tall sponges.
I like how they have little things even growing on top of them. I don't know if that's like a bryozoan or like maybe like
Got
another little.
Narella corals. We've got probably some hemicorallium here.
And it's been active for
Branch
so
bamboos.
long. Look at these dead ones. Happy things growing here at Coral Knoll for a long time.
Some nice angular material here too.
Okay, look how big that coral is in the Atalanta view.
That's got to be the size of like, that's multiple dinner plates. That's ginormous.
Yeah, that's a nodal brancher also.
Well, it's interesting. They're usually only see them on one side, but there's a lot on the other side of the knoll here.
This might be a good place to grab another rock as well. Looks nice.
Yeah, I could grab one of these here. There's a little landing zone here.
Yeah, maybe this guy?
Sure.
Actually, yeah, if you set down now, maybe we can do a twofer, because there's another coral I want in this area too, off to our lower left, just outside of the frame.
You want to circle him? So.
It's,
Oh yeah, go ahead.
yeah.
I'm trying to think of a good analog for something that's like a meter
It's
tall or a meter wide.
Still more back here.
Hula hoop.
Thank you.
Which then, I'm like, here we are, spending
Yeah, it's right in front of your
$1,000 to make
toes.
hula
You can
hoop
see it in the
for
DSC. It kind of looks
fridge.
like, it's right here.
Roger.
Those are both good ones.
This one here.
Or we could start getting into like the cars.
So
you
maybe
know,
we can do
like,
a twofer here.
Yeah.
Is this a Mini Cooper?
A couple of these look
Sample
like
that
they're
right
not
here.
Smart car.
Yeah, we don't need any of these, so this is
Sorry,
a known
Dan.
species, but I'm interested in getting a small piece of this one.
Roger.
That dead sponge looks like a vertebrae.
It does. Those are those rib cage sponges. I don't think
It's
it's their
actually
real
in the
name.
DSC there, is it?
Yeah, I got it. I'll do another one.
Oh, back up, Herc, back up.
Is that exposure okay?
Oh yeah, that's great. Yep. It's hard. I've been having some problems getting the DSC to focus on the entire frame. It usually the edges you have to crop out because they're out of focus.
Hello, shrimp. Oh, that's a cool picture.
I'm just making sure these rocks aren't too
Yeah,
close.
quick zoom on the shrimp while he swims into the frame there.
Betcha.
I'll get them in about. I'm gonna do this
Nice.
relay isolation dance here again.
It's doggy paddling. Just like me.
Wow, that's wonderful. It's such a methodical way of moving his, would you call those feet?
Swimmerettes is what they're called.
Swimmerettes.
Really?
Swimmerettes.
That's cool.
Yeah.
No idea.
A shrimp swimmeretting past.
Not pirouetting.
Can you do 5 for me, please?
5.
Or if you want to be
Oh,
scientific.
he's hanging up by the
pleopods.
arms. We got him on the bubble.
What is it, Steve?
Pleopods.
Pleopods.
It's also pretty cool.
We get full
good
zoom
word.
there. Or just,
Pleopod,
maybe
pleopod,
not
pleopod.
quite full.
Yeah,
I can't say that 5 times fast.
Pleopods?
Yeah.
Pleopods.
Geopod.
Yeah. So this I believe is Calyptrophora, the genus, but an
Can you
unknown.
move the bubble, chase the
Oh,
end of the
yeah.
manipulator?
Can we zoom in right here for a second?
Sure.
This is something that I've seen before that I wanted. I didn't see this when I first sat down, but now I'm very curious
Smacks.
about what's going on right here in that little gall.
It's full zoom
Okay,
there.
so we're looking
Oh,
at the
wow,
white
look
the
at
white
this
lump in between the branches.
Brittle star too. Yeah, look.
Kind
Are we
of-
full zoom?
Yeah.
Yeah.
Oh, okay. Do you think you can grab this somewhere, whether you take a branch like that or
Absolutely.
take a branch like that?
Can you show me that coral with bubble cam?
What are you thinking it might be?
So I think it's a gall of some type. So probably a parasite
there?
that attached and
Right.
was recolonized by the sponge. And I've seen these before. Actually,
Do me a favor, Dan,
on
zoom out just a little bit. That's good, thanks.
last year. I don't know which cruise
This
it was
is a
last
cool
year.
shot
That
when the manipulator's
too many
Okay.
cruises last year to keep straight. But on primnoids, sometimes when they overgrow, they create kind of a random arrangement of sclerites around this, parasite, and it forms a gall.
Is that the coral?
And I've never really understood how or why they do that, or if there's any difference in the sclerite morphology that are on these galls versus what are in the polyps.
Yeah, I got you.
So I'm really interested in getting a piece of that if possible.
So this thing just built a like skeleton cage around something that wounded it, and just was like, here's what I've got to build with, bones,
Yeah.
you know,
Cool.
and just like packed, surrounded it.
I'm going to encase you in my, my armor.
Inner armor, and then just like went on,
Yeah.
kept on, keeping on.
Yep.
I'm just gonna.
That's pretty cool.
We can take this is a fairly small colony, so we can take, a cut like here
Roger.
if you want.
Can we make this a highlight? A number
Yes.
5 highlight? So this is a ham shot, so you have the fully zoomed in video and then dun, 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 dun.
You have to add the music to the highlight.
Totally.
Right.
We need Dan to direct the next Friday the 13th.
So the spelling on this one is, oh, you got it right. Good. Cool.
I'm giving it 4 stars.
This is assuming I don't choke on the.
I believe in you.
There might be a second gall, in fact, too. Like both of these might be.
Yeah.
Yeah, it is one of those galls. I can see it now.
Right enough there, Steve?
Yeah, plenty. Yeah. Beautiful.
Awesome.
There's a really good, it's oftentimes for genetics, all we need is like 10 centimeters or so, or an even smaller snip. But oftentimes, there are some important morphological characteristics like branching sequence and pattern that we can only really get from understanding the larger colony morphology too. Yeah, nice photos. Yeah, that's totally one of those galls. All right, we're all set back here.
Okay, you can go right now. That's going in the front too, I'm assuming?
Yeah, we can do that. Yep.
Front right or front left?
Same compartment.
Same compartment as the other one?
Yeah.
Roger that.
Really quickly answering that question in chat. Have you discovered anything new or multiple things new since you've started this expedition? We believe so, but we won't really know until more after the expedition when we send in our data to our labs onshore.
Yeah, I think it's just as exciting to have range expansions, and we know we've had a few of those. A range expansion is when, we find something that we know, but we didn't know it lived here.
Yeah.
And here either being, there's a lot of scales of here, right? Here could be a new depth. It could be a new area, the Central Pacific. It could be within the Johnston Management Unit of the Pacific Remote Islands Marine National Monument, which means
you might have to open some
we're
more.
protecting species that are here from future impacts, which is exciting to expand what a manager might call a species inventory, a list of who lives here. But I don't know, for reviewers, I get just as excited about that, right? It'll take years and years and years to know if it's new or if it's a new species, a new subspecies, a new
still got a piece
genus.
of it in there.
But like right away, we get the results for knowing that things have been spotted in a new area.
I think one of our scientists ashore was saying tripod fish maybe was a new location for it.
Yeah, maybe need
a couple days ago.
Oh my gosh.
That's awesome.
Oh.
That's all right.
It's okay. 4 bucks for...
We can make a note of that. Yeah,
We'll have
broken
a
branch fell into
It might have
Omega.
gone in the other one, I can't tell which one.
It's amazing.
All right,
All right.
rock. Yeah.
Yes.
All right, Steve, folks want to
Somewhere on the
know.
right side of the
Okay.
screen, I think I
You
saw.
go
That is
wide
sample
there. See any rocks you like
075.
there? So many.
Wait. Well, wait. We want
What was
Brendan
that?
to get his rock, and then we'll go
See
back
any
to the
big
question.
rocks you like there? We're kind of light.
Look
Use a
in.
big rock. But any rock you want. If you
Right.
circle the rock, I'll pick it up.
Some of them look a little thin.
Eeny, meeny. So,
See any thick ones?
eeny,
Please be careful about shaking it in
eeny, miny.
Yeah, let's, maybe this one?
I was a finger on the button the whole time.
That one, maybe?
That one?
Yeah, try it, yeah. It's hard to telling how, you know, how big
Making
it
the
is,
boss nervous.
but
That one is
stuck? Okay.
see if I can get it out of there.
When we collect the sample, Brendan, we have a
Negative on that
question
one.
for
Okay,
you in the
let's
hole.
do this. Looks nice and blocky.
Yeah, that one's loose.
I think it's jiggly.
Jiggle, jiggle.
Go to grip force 9.
What grip force do you use for the coral collection?
3 or 4. Look at that finger grab. Oh my god.
Yeah, that's nice. Me likey.
So tell us what makes a great sample for your research, Brendan.
Well, I feel like I'm repeating myself, but
That's all right. New folks online all the time.
that's true. Not 100% sure yet, but I want to see a good amount of volume for these rocks in terms of all 3 dimensions in volume, as well as angular edges to it, and I see both of those here, so potentially good for research.
So, Brendan, someone in the chat says, rocks, all the rocks here look like magma rocks. Is that just the area you're in right now, or is most of the ocean floor made up of magma rocks?
¿Por qué no los
Go
dos?
for full zoom.
For those of
right
us
there
that
if you
don't
want.
speak Spanish, what does that mean?
Why not both?
Hit
Same.
5. Hit
Yep.
5 for me.
The area we're in is known for magma rocks, as well as most of the ocean, actually, almost the entirety of the ocean floor is magma rocks.
Oh. Are there any other kinds of rocks, or is it just solely magma?
Okay,
No,
coming out.
you can find fine-grained rocks as well.
Huh?
See, this should be A, B, C, right?
Mudstone, siltstone,
Yeah, you can put it in C or D.
shale.
You're happy with C?
Yeah, C is fine.
But yes, mostly magma rocks. That's how the ocean. That's how the crust forms through magma processes.
Nice one, Dan.
Beautiful.
Beautiful.
Really good.
Can I hit
Now that
that
that's
again?
done, we can go back to picking out our nice shag carpet here.
Yeah. What are we looking at, Steve?
About 5 there, is it?
A bunch of sponge spicules.
No, it's not 5.
Dead sponge.
Is it
That
like
gives me the
a chunk
reference
of
there.
a sponge,
Gotcha.
or is this just spicules that have
Beat
fallen
myself.
apart? Like a
Yeah,
puddle?
they're probably old bases of some of these sponges. That oftentimes for the polyopagon sponges, the base material kind of has this texture where it attaches. It's probably that, but it looks like it's collected here. They're most likely fallen from above, maybe.
Ah.
Awesome. That's awesome news.
So, Steve, someone in the chat wants to know, what exactly are sclerites? A sort of tissue?
Sclerites?
Sclerites, yes. A sort of tissue? What are they?
They are shards of calcium carbonate, mineral.
They are the
Every
I could hit
species
one on the,
typically produces different, unique morphologies. All of the different morphologies of sclerites in a species is called the sclerome, kind of like genome, but sclerome. And so by comparing all the morphologies, you could tell back in the olden days, and also still today, that was the only way to tell species apart. But now we like to use a combination of different morphologies to tell apart different species, which includes a genetic component.
Okay, Dan, you can go in.
Good collections.
Yeah, that was
Great site.
How awesome would it be if
fantastic.
Coral Ridge just like stays this way, the whole coral knows the whole way up?
We wouldn't get very far.
Yeah.
That's awesome.
Yeah,
That's what I'm worried about. Yeah.
Spend the whole watch here.
But it would be awesome.
I wouldn't be mad.
We haven't gone up and down the cliff yet, and there's the whole other side.
think we've
All
barely
we've looked at
moved.
is like the top of the wall.
Jess was right when she said we'd enjoy this spot.
Yeah.
Good thing you found it with Atalanta. I would have
Got real close.
That's why it's always nice to have two ROVs. Oh yeah, we're a little bit heavier now. Sweet, this is about where we should be.
This is great.
We dropped any weight yet?
They must have.
Yeah, we dropped 2.
Because
These don't
Look at
really
these
look
big
like
giant-
erupted features. It's probably a failure.
Yeah,
Yeah.
that'd be my guess.
What kind of failures happen on seamounts like this?
landslide.
Gravitational failures.
Gravitational.
That sounds way worse than it actually is.
Okay.
That sounds like the universe is collapsing.
Speaking of the universe
Can we,
collapsing,
yeah, can we predict when those happen?
When's the big one going to come to Pacific
I don't think
Northwest?
so.
That's what I want to know.
As with most science questions, though,
Right.
you can always say, well, how much money do you have?
So there is a field of geology studying that kind of thing. You can use, we call them PBRs, or precariously balanced rocks.
Okay.
And you can calculate using, its dimensions to figure out what magnitude of earthquake it would take to remove that rock from its position.
Okay.
And then you'd be like, okay, this rock is this amount of years old, it hasn't moved, so then there hasn't been an earthquake of that magnitude in that amount of time. But deep ocean, I think, is a bit different. Perhaps you could do it, but I don't think anybody's tried.
That's one of the things that I'm very interested in from a seamount ecology perspective is, the seamounts are not immune from earthquakes, even though we're not on a plate boundary necessarily.
Yeah.
What would be the effect of different magnitude earthquakes on the seamount environment? Would they cause like turbidity flows and benthic storms and things, or would they result in failures? Yeah, I've always wondered that. I know spent a fair bit of time doing exploration in the Caribbean, in the Mona Passage.
I think it's a bear wreck.
And in the Mona Passage, there's a place called Mona Canyon that is on a very tectonically active area of the Northeast Caribbean. And
Zoom in real quick there, Dan.
failures happen there all the time, and nothing can attach to the walls because it's so frequent, like corals and sponges, so it's barren.
Because of the seismic activity?
Yeah, because the carbonate fails so frequently, presumably.
Wow.
Breaks off.
Yep.
Look at that cousin it, hold fast.
But yeah, it's
Okay, you can go in.
So cool.
it's not unusual to have
So what is that,
earthquakes.
Steve? That fuzzy...
Oh, that's a sponge base.
Yeah.
So that's what I
Yeah.
was talking about before, the shag carpet.
Not a shack.
Yeah.
Next.
To get a large magnitude earthquake in the middle of a plate is extremely rare. Rare? Yeah. It's possible, but
just not common.
because slip on tectonic plates is what causes an earthquake.
It's going to pop down the
And
cliff here
well,
one
we're not
way
between two
before we
plates,
go.
so
Yeah, if you're a longtime Nautilus fan, you'll know some of that work we did was looking at benthic geohazards in the Caribbean.
Oh.
And we also did a lot of work in the canyons on the west coast, which is where you have, the meteor kept Cascadia subduction event, and we had a lot of conversation then about it's a really different environment. It's a super sediment heavy environment, but how, you know, both turbidity flows, which are just mud becoming heavy and clumped together and running downhill like a mudslide, or how, you know, maybe earthquakes or small tremors would release sediment there and change a canyon community.
Actually, speaking of earthquakes and stuff, usually during an earthquake, do we see a lot of like magma, or do you see a lot of stuff on the ocean floor here?
During an earthquake?
Yeah, or like when a tectonic slip happens.
Like what? Like geologic processes
Yeah.
result? Yeah, you can have a magma release.
And then
There's only
going
like
into
a
that,
couple
quick run
witnessed
over the
times
graveyard
ever in
here.
the deep sea
Always
to actually like
find
be
these
there with an
interesting.
ROV and see them. One was in the Caribbean, which is so cool.
you saw an earthquake?
Not us, not Nautilus. I mean, like that's how rare. Like all of humans have only ever witnessed these kind of events a couple times. There's like a couple instances of underwater volcanic eruptions with an ROV being there over in the Marianas Arc. And they're recently in the Caribbean, in the Puerto Rico Trench, there are events there that are dominated by shrimp. And while I believe a Woods Hole team was there, I think it was Jason was diving, and like this whole pillar of shrimp are just like going about their life, being shrimp. And all of a sudden they like all leap off the substrate, and that was like timed back and triangulated that there was an earthquake and a slip, and like you see all of the animals on the seafloor react and come off the seafloor. It's just like you could not, like, that is just pure luck.
That's
that
amazing.
is like pure
It's really cool.
you know, we're talking about things that happen like in the million-year time frame. Happen to have a robot in the right spot in the right time.
I'm sort of picturing like just before an earthquake or like a volcanic eruption, all the birds take off an island in the movie, you know?
Yeah.
Oh, yeah.
It's that, but shrimp.
But shrimp and underwater.
Yeah.
Even cooler.
Super cool.
So Brendan, somebody has a question specifically for you. Question. If we have only mapped 20% of the ocean floor and visually laid eyes on 5% of the ocean floor, how can one say the majority of the ocean floor is magma rock?
Well, I would argue we have less than 20% and less than 5% on both those parameters. But I mean, it's impossible to see everything, right? But we know the processes that occur, and it's logical to assume that they occur pervasively. Now, I mean, I appreciate the attempt
You're
to,
good for a GBL reset.
question that, and I think that's a good thought process. But
however,
I mean, I can't know for sure. You're right. I can't tell you if I know for sure, but that's what makes sense, and that's what everybody else thinks as well.
Yeah, we talk about this a lot. I think there's kind of like these scales, right, of like what does it mean to have explored a place?
Suck you in.
Right, we're gonna get
The current
this incredible detailed view of this place,
blowing up the cliff
but we're
face.
kind of like walking up a
That's what
mountain,
happened
like
there.
What is the current doing?
hiking up a mountain with a flashlight, right? We're gonna
I'm
see
not
the
sure,
very
but
specific
I
ridge we
definitely
saw,
got too close there
and we're gonna have
sucked
seen
me
it
in.
at this time of year, right? So we know
North,
how
south,
stable the
somewhere.
deep sea is by what we have studied in these few remote locations. So
Oh, I could stay here
maybe it is
all
unlikely
night
that it would
long.
be different like fall, spring, summer, winter, you know, but you could put a temporal aspect to exploration and say like, you've been here once, have you really
That's not
explored it?
accurate.
But also I think like, it's so exciting to think on like that big scale of like, okay, so if we visit a variety of sites at a variety of depths.
Okay, we're ready. No,
We
actually,
can say
no, we're
something.
going.
We can't say everything about the place.
I lost the plot. It's been so long since we moved, I forgot
Sometimes
which
that's
way
all
we're
you can
going.
do as well.
Yeah, totally. Like
You have
Southwest.
no choice but to explore what you can and then apply that to other
that
things.
works for me.
That is ginormous.
Wow.
Before we do move, there is, do you want to look at the other side of this feature at all, or you just want to get on with it?
Those are great.
Other side, like to the left?
Yeah, just over the top here, there was.
Yeah, we can take a quick peek. I really do think there's more better stuff coming up.
more
amazing.
better.
Well, we better get going then.
Are the lasers on?
Go ahead with it.
Yeah.
They are.
So
Sweet.
why don't we move onward, since
Oh yeah, I can see.
there's more slope coming up.
So speaking of the lasers,
Unbelievable.
somebody in chat wants to know how far apart of the how far apart are the lasers from each other,
Those are 10 centimeters.
or 4 inches for those of us in the U.S.
Thank you.
Gibson, you have excelled at the ROV intern question. That is the go-to first question for
Is
all
it?
ROV
Yeah.
engineering interns, Dan.
I got it.
Congratulations, Gibson.
5
Thank
watch
you.
bucks.
Yes.
Yeah.
Forgot about the watch box.
And then another one. What is a benthic storm?
It's when
Amazing
the
band
ROV
name.
makes a bad landing.
It's the name of my next punk band.
Yeah.
It's a good band name.
It's a good
Benthic
ROV
storms.
name.
Yes.
Benthic storm.
Benthic storm.
Totally.
You want some more leash
If
there?
I ever have a research vessel, it's going to
Come down a
call
bit,
Benthic
yeah.
Storm.
Okay.
I
Down
love
10 meters.
Coming down 10.
Yeah. Basically, whenever you have instabilities in usually sedimented slopes, most commonly in things like submarine canyons, typically off the continental shelf. Also, some things, they can be triggered by, I don't know, usually something tectonic, like maybe earthquakes or I don't know what else. But yeah, so what happens is you'll typically have a failure of the slope where the sediment, which is usually fine grain and usually has a very high water volume, liquefies and turns into a slurry that kind of like an avalanche.
What is the difference between a turbidity flow and a benthic storm?
I think that they're the same thing.
Synonyms. Sweet.
That's a good watch name.
I love it.
For those of us just joining, hello. We are just going over this unnamed seamount near the Johnson Atoll, finding a bunch of sponges, a bunch of corals. It's a really nice time.
Do a zoom there if you want, Dan.
Yeah, I'd
Bring
encourage
up
you to hit
squad
the
in
refresh button if you haven't in the last
Squad
2
lobster.
hours. You'll see our watch team pop up.
there. Yeah, you can go in tight. if you
Sure.
want.
And then to
Look
answer
at
that,
that. That's
oh
so great.
wow, is that another squat lobster or is it a different kind of crab?
That is a type of squat lobster.
Ah,
That's called a galatheid crab, different family from the ones that we usually see. Might be Enidopsis, possibly.
That's so cool. That is also going in my calendar of images from this expedition.
and then to answer the question of how much longer is on this dive, we're not entirely sure because we had some technical difficulties at the beginning of it, but it's somewhere between I believe the next 12 to 4 watch. Megan, would that be around there?
Let me see. Dive was planned to be a 18-hour dive. We ended up getting it back in the water
Okay,
around
thanks.
10 this morning. So yeah, probably midnight.
Go,
I don't know. Our team,
I'm waiting for the
Yeah, I
boat
think it's
there.
going to be around
Yeah,
midnight.
it depends on our progress.
There's
There, I think we can.
And we have too many cool things to look at. Going
That's
we
not
are
a bad
moving the
thing.
boat, right?
Yeah, you want me to keep moving?
Yeah.
All right.
All right, ready?
Bridge nav. Same step, please. 20 meters, 255.
So Steve, what are these little hairs all poking out from this sponge?
Spicules, glass spicules. Yep, glass fibers.
And what are they used for? Are they for defense, for gaining food?
Not clear. They're mostly structural, but they may have other purposes, but primarily structural.
All right,
Okay.
is the spicule to the sponge, the sclerite to the coral? Is that
Right.
the right comparison?
Yeah, yeah.
Will you be my spicule to my sponge?
But they can also, you know, we can have sponges also. The spicules are usually, the glass spicules are unique to the glass sponges, the hexactinellids, but there can be
Is that a
different
crinoid?
sponges that have
Is this
different compositions from
Featherstone?
calcium carbonate to protein too.
Wow.
Is it a sea star or a crinoid swimming?
It's a swimming crinoid.
Oh my lord!
Love
That's
it.
a good another good band name, the Swimming Crinoids.
You like that better than the benthic crinoids?
The benthic storm, you mean?
Yeah, the benthic storm.
I mean,
I'm just saying, if you like benthic, you can put it in with crinoids.
The benthic crinoids.
You pick your favorite one.
Yeah.
I still
Walking
prefer
through water.
benthic storm.
Here's an
Flying
embarrassing
band.
question about sponges. Is a glass sponge, is it actually glass?
No such thing as an embarrassing question.
I was thinking the same thing.
I want to say yes.
Okay. That's a good
Yes.
question.
Okay, makes me feel better that a lot of people don't know.
I was wondering.
yeah, I
the
have no
same thing.
idea.
They feel like glass when you, they feel like fiberglass when you touch them.
Okay.
Yeah, the spicules are silica dioxide, usually, you think? And that's silica's your same, yeah, your car windshield, your windows in your house, your any of it.
Awesome.
Speaking of sponges, Steve, we have a question. How common is it for sponges to have external/exposed spicules?
Pretty common. Yep. Yeah, they can be some part of the structure of the sponges. Sometimes they
Keep
can be
moving?
left over.
Yeah, keep
Yeah.
it moving.
All right.
Yeah,
But
it's keep it
Atlanta
moving.
hasn't started moving yet.
Like you often see these
Lobster.
exposed at the base here. These exposed spicules as well.
Yeah, they're kind of like anchors. Really cool.
Good for a tight zoom there as well. We can get
More
one of the little
Galatheid crabs?
critters there.
Is that right?
Yeah, yeah,
Awesome.
Galatheids. Yep.
It's Max.
Look at that mesh, right? Like, how incredible is that lattice?
It's called there's a terminology for it. It's called a sieve plate.
A sieve plate.
Yep.
Awesome.
So does the sieve plate kind of work like an actual sieve? Like it just all of the detritus comes in and then it feeds off of it and sorts it out?
Actually, this is the X current side. Usually the sieve plates cover the X current, so where the water's coming out of the sponge.
Oh.
So the water will typically come into the sides and out through the top of the osculum, which is in a lot of glass sponges is covered by a sieve plate. So yeah, you're kind of looking at the down current side here.
It's like the last chance to catch anything that's
Yeah.
filtering through.
Or just to block things that might clog the flow.
Mm-hmm.
That would be pretty detrimental to the sponge if it wasn't able to pump.
And so the spicules are overlying the cells, is that right? The oscula and ostia?
Yes, yeah, in this case. But they also make up the composition, the mass of the sponge too.
Okay.
Yep.
Does the sponge have any way to influence the flow of water through it, or is it just passive?
Yeah, there are small cells that are inside the sponge that create a current
Wow.
by.
It's like a lung.
Yeah, yeah.
Yeah.
in a way.
Sponge lung.
So yeah, they also can do some pretty cool stuff where they can clear out sediment buildup, or they
Wow.
reverse flow, and
Really?
Yep.
Oh my gosh.
I think there's some video of that on
I
the
call them
internet
like
somewhere.
sneezing.
Yeah, sponge sneezing.
That's amazing.
Using sponge.
I don't know if that applies to these specifically, but
Yeah, I don't know.
yeah.
And there's sponges with other kinds of composition on it, right? Like demosponge sponges, and is there other types of sponges out there, and what are they?
Okay.
The demo sponges, the glass sponges, I don't know too much about sponge diversity. There are calcareous sponges too that are made out of calcium carbonate. Most of what we see here at these depths are glass sponges, and to a lesser degree some demo sponges, specifically the carnivorous sponges.
I thought it was usually the concave side of a sponge that was the inlet side. Do I have it backward, or does it vary by species?
It depends on their morphology.
Come down 10 meters.
Typically with these
Coming down 10.
with these sponges, I think it's the opposite
Okay.
from what I've heard from Chris Kelly.
Okay.
But you know, it's not like someone's down here with dye, you know,
Right. Yeah.
doing tracer experiments,
Which we have done with fluorescein.
which we have done.
We have?
Yeah.
Yeah.
Good to know.
Also done in shallow water. It's
Yeah, shallow water
spectacular
stuff.
in shallow water.
Never done it in deep water.
You can find it on YouTube.
I can barely hear you back there, Megan.
Oh, really?
Yeah, you're real faint. What station are you at?
I'm at the chart table. Is that better? I'm super
Yeah,
loud in my own
I
ear,
don't think
so
I have
I'll
a
turn
table
myself
button
down.
as well.
Yeah, that
Oh,
station
you
is
don't have
very
a trainer.
loud.
You're right.
What's
Okay.
that?
It's
Oh,
the
How about-
red trainer for me.
How about now?
now,
Now you're fine.
yeah, I clicked you on. Now you're
Okay.
Oh, I see. Table.
table.
Oh.
So what can you tell us about these crabs? Are they helping the sponges? Do they live there all the time?
Yes, ma'am. Yeah, keep it. Keep
I
going.
don't know, honestly.
Steve wants to cover some ground. So.
Yeah, I would imagine they're probably using it as some sort of shelter. I don't know if they contribute significantly to the process of the sponge, but it's a heck of a lot better than being exposed to predators and things
Yeah.
like that.
We've seen a lot of them for sure. And again, I think I feel like the headline of so many of these creatures is like, get out of the benthic boundary layer where the water flow is like tumbling across all the rocks. Like the higher you can climb, the more food you can get, you know, is really across animal group, like a really good strategy that we see lots of these things playing out.
There a touch
There's
first.
another euplectellid here, I think. So that sieve plate is now on the top, as you can see. That's the osculum. This one looks a little bit tortured. It doesn't look like a typical euplectellid, kind of one of these vase sponge morphologies. They can grow in a lot of different morphologies, which makes it very difficult to identify sponges
Just gonna do a
from
quick check on the
video or
gauges.
photo alone.
Roger.
So typically they are unidentifiable to species unless a specimen is collected to look at the sclerites or spicules.
So Steve, why do you suppose you see, someone in the chat wants to know, why do you suppose you see mostly glass sponges at this depth and not demi-sponges? Are glass spiculars are stronger?
I don't know in this case if there's a kind of evolutionary reason. You do see demo sponges this deep, but they typically have similar traits in some cases. They're usually translucent or you know, very pale or white, much like the glass sponges. But they might have different life traits, you know. They might, in this case, the carnivorous sponges obviously are predatory, but not actively predatory. So that may be a benefit up to them as coming from the demo sponges. But the glass sponges, I'm not sure what their why they would be better suited for this environment. I imagine it's probably in part due to the abundance of silica that's usually present in deep waters. Oh, there's a nice Pharea. Another type of sponge. I don't think we've seen this one yet.
Zoom in there,
Genus
yeah.
Pharea, it's a glass
Have
sponge.
zoom for us.
These look like bouquets to me, like bouquets of flowers, just the layers and the intricacy.
Yep.
Hold that.
Yeah, so I would imagine silica is a good construction material since it's widely available in the deep waters.
Okay.
And it's persistent. It doesn't readily dissolve like calcium carbonate might. You know, if you've noticed, a lot of what we see is sponge debris and not a lot of coral debris, even though we see massive corals here. Maybe you might see some bamboo coral debris, but over time, the coral debris will dissolve because it's made of calcium carbonate. And we are well below the calcium compensation depth for, especially for aragonite. I think calcite's a little bit deeper, but still, you know, things are not going to persist in this environment very long if they're not, in this case, corals protected by tissue. The tissue of the corals protects it from being attacked, so to speak, by the relatively
Checks are good.
low
Just
pH,
good.
lower pH seawater.
Attacked by chemistry.
Attacked by chemistry.
I think there's some students who would also feel like they have been attacked by chemistry.
I second that notion.
I third that.
Chemistry only loves. You just weren't willing.
I actually have a minor in chemistry, so chemistry and I are cool. We're cool with each other.
Excellent.
You're well bonded.
And with that, I'm going to go.
-5 watch box.
I didn't know that was possible.
we just,
Be thankful it's only 5.
we're just starting out too.
I went up and down and back at 5.
Oh.
That's kind of
No,
an
you can-
interesting little, how they've colonized just the very ridge there.
Yeah, that's pretty neat.
It's a really thick stem on that coral right there.
Yeah.
That's incredible, right? You can imagine it's been there a long time. So there are glass sponges displayed in museums, certainly. They're challenging to preserve from our science team because they
Roger.
it's really hard to dry them out because they have a lot of tissue and a lot of like really dense just structure to them that can make it really hard to get them totally dried or totally preserved to get
Look at the starfish
alcohol
there.
in, replacing everything. Oh yeah,
What a beautiful star.
nice star.
Oh, look, there's a slime star as well, I think.
Yeah, it'll be interesting to look closer. Meredith, how's your sea star ID?
My sea star ID, not great. Okay.
It's got 5 limbs, so it's a sea star.
You can,
Ta-da!
Oh,
Zoom
we're looking
on
at
the
that one back there?
slime star.
Yeah.
Yeah.
I think that's a cushion star.
Yeah.
Okay.
Thank you.
Oh yeah, that's a cushion star, maybe.
Dr. Christopher Ma is really one of the go-to experts on sea star ID. He's at the Smithsonian and really active on Twitter, so we appreciate all of his support when we
Okay.
are searching for good IDs on
Don't
these.
mind.
Kind of looks like a belly button.
If that is indeed a cushion star, that's where the mucus comes from. Yeah.
Yeesh.
It's good strategy.
They're also called slime stars.
Oh, I was right.
Yeah, it's a super, super big group.
I said, Dan's piloting, he won't be able to eat. And she's like, he'll be able to eat it.
I would let you in here today, Chris, but there's no way I'm getting out of this seat right now. Sorry.
too good.
But after I just had that collateral damage, I probably should.
It's a good question. We've had a few questions about pollution in the marine environment. We've definitely spotted some big
Yeah, we're
plastics.
stepping. It's going to get really steep here.
Taylor Ann Smith, our science manager's personal research, her graduate school research is about microplastics in the deep. I don't know that we're collecting any samples for Taylor Ann on this particular expedition, but we have collected samples
Actually,
in the back. Ooh, she's walking in for dinner leaf. We will have a chance to
behind the curve here.
ask them all about it.
Well, I think if I get on with it.
Got some beautiful sponges and some black corals.
So Kevin, tell us about this rock formation here. This looks really different than a lot of the slopes that we've been flying up.
Yeah, we're seeing some in this seamount really thick basalt flows. So when you have a really thick flow, you can get these large blocky features. We saw some columnar jointing earlier, really like radial rose pattern, really beautiful. And so I think that's what we're seeing up here. It's just kind of really thick lava flows.
Is that just like a lot of lava coming out all at once?
Yeah, essentially.
Okay.
It's just kind of inflating in itself for a long period of time. You can find in some islands, like I was on, let's see, Tahiti Nui, and there was like 6 or 4 meter thick lava flow. Sometimes you just get massive flows. Yeah.
Is that just from like just out of chance, or is there positions on the tectonic plates?
It's not necessarily due to tectonic. It's more localized. It's just, yeah, for some reason you have a large upwelling at one point, and you can get a really thick lava flow coming out. I don't know enough about the detailed volcanology to say why exactly, but you do see them. These look like really nice rocks. Looks like you grabbed a couple. So that's good.
Hey Taylor, you arrived at the perfect time because viewers want to know about plastic residue in the deep sea. What do we know about it? Are we testing for it on this expedition? Tell us everything.
Yeah, well, you're lucky I just walked up here. That's
Yes, we
actually
are.
my research focus is microplastic pollution in the deep sea. And yes, we have seen plastic at these depths, even deeper.
No, it's...
I feel like every cruise that I've been on in the last 4 years, I have seen a piece of plastic, at least on one dive, on my watches. But yeah, research indicates that microplastics are ubiquitous within marine environments. So yes, I've tested and collected samples from within the Monterey Bay and Channel Islands National Marine Sanctuaries, and I'm currently researching and doing data analysis on the results that I have, where I have found some plastic fibers made out of polyester and nylon, some things like rayon, which are mixed with cellulose. But yeah, we do know that there are microplastics in the deep sea. So part of my research is trying to figure out what types and abundances of microplastics exist, not only in the deep sea, but along a depth gradient from the surface to these depths, and what types of microplastics we're seeing, and if they are found inside of the organisms and their environments. So I've collected sea pigs and some sea pens and urchins to see if there are different feeding mechanisms, if they're ingesting microplastics, even though they feed differently. So if these sea pens could be grabbing microplastics from the water column where they feed, and if these detritivores could be feeding on it in the sediment. So I would take push cores and water samples paired along with these collections to see and compare to the environment. So right now I'm at that phase of finishing up those final lab samples and doing data analysis to see what significance results I have.
Can you tell us some of your results that you found out so far?
So far, I'm not sure if anything is statistically significant or not yet. I'm still working on that. But yeah, I found mostly just microplastic fibers. I haven't seen any microplastic fragments in my samples, but we're really, really far down in the deep sea. And a lot of the samples that I have And I feel like it's a lot easier to potentially just find smaller things here than really large pieces of plastic inside of these organisms. But yeah, I have seen polyester, but a lot of the things I have seen are natural as well, that look like they could be plastic fibers, but they're not. And also I have to account for contamination and make sure that the samples that I'm looking at aren't contaminated. So I have what you would call blanks to kind of offset that and subtract those fibers and plastics that could be due to contamination.
So what are some of those fibers that you said there's some naturally occurring fibers inside of these animals that trick you for plastic? What kind of fibers are those, or what kind of materials?
Yeah, so there are these fibers that are mixed with like cellulose. So there are things that are somewhat natural and somewhat not. It's like a mixture. And I have been doing more research on the different types of plastics that come from where. So this way we can make informed decisions and, give results to people that, are making more decisions on our policies. So we can start moving away from using certain materials if we are using them a lot and we're seeing that for some reason they're, finding their way in the deep sea more or than any other type of plastic. So it's good to know what types of plastics we have so we can potentially find the source of these plastics so we can stop polluting, which that's going to take time for us to, move to more sustainable resources, especially in our own work, which is something that I would love to be a part of doing. Yeah, and I think it just takes a lot of collaboration from different folks, from scientists, policymakers, engineers. Everybody needs to be a part of this type of research in order to solve such a wicked problem.
So what inspired you to start doing this?
Actually, my first year on the Nautilus, I heard about somebody doing microplastics research, but not in the same ecosystem. So I was interested in knowing a little bit more about that.
It never fails. I take over from Steve, and all the corals disappear.
It's true. We were seeing
Oh,
a
man.
ton before you got here.
Yeah, we were in like a sponge and coral garden for a while with those big boulders.
Can we get a little zoom on that coral that's just at the bottom of the screen? My timing was off there. Tapping in. Nice little bamboo coral.
Can I get a zoom? I get it.
Thank you.
Okay, come wide. Thank you.
So it looks like we've got this bigger flat area to kind of cross, and then we'll get to another steep section before the next waypoint.
Halothurian at the bottom.
So what kind of ice cream did everybody go for?
The macadamia vanilla one. Not a big coffee fan.
I'm hoping there's some left when I go back down there.
There will be the macadamian for sure.
Challenge accepted.
And you'll put some strawberries with it. Ooh.
What I'm trying to figure out is why are all the controls up here sticky?
Sling another halothurian.
We were meant to do this on every watch change anyways, but we haven't been. Are those ones all right over there?
Yeah.
Totally slimed them yesterday with a peanut butter bar.
You're touching it.
Oh, sorry.
Thank you.
We never did do that ground
It always
Where?
used to, but not on the previous trips this year.
We had a ground wire on it.
Right, but it always used to do that.
So the manip runaway thing
Yeah.
was lost comms.
Nice little anemone.
You know what kind?
Well, I will take a look.
Can I get a zoom?
Of course.
I'll take more.
It's some sort of Actinostolidae, but they're not. We have a family, but no good species references for these guys.
Okay, come wide. Thank you.
A nice little shrimp there behind it,
Full
posing.
wide.
Big chillin'.
Photo bombing.
What button? Oh, yes, that's what, I don't use it either, but that is, I heard that from Josh yesterday and Jess today. Yeah, yeah. Or I just pinch it the whole time, which, and index, me too, yeah.
Can we get a zoom on that little coral that's right at the lasers?
Yeah, Chrysigorgia.
Yeah.
Can I get a zoom?
Zooming in.
Thank you.
You're welcome. Is that a squat lobster in there?
I think it is. Hanging out in the branches.
Oh, come wide, please.
Coming wide.
Thank you.
We have a question in the chat for our ROV people.
Sure.
What was the problem with Atlanta? What was it fix? What was the fix?
I was asleep, Dan. What was the... I know. I saw the message at first, and I was like, oh, no. And then I said, it's back in the water. I said, oh, yeah. Good job. That's okay. It scrambles my brain. The frequency. I can't wait until I am. Oh, okay.
You're not on SPL.
Womp.
I'm sure he's got a great story going.
Sorry. Yeah, the problem with Atalanta, we lost telemetry during the descent, and it turned out to be one of the fiber optic cables that all of our communication, video commands, all that stuff goes to. One single little tiny glass cable. And there's a couple of them on the vehicle that are exposed to seawater, so they come out of the main electronics bottle and kind of run
Maybe grab this rock.
through the vehicle.
You want to grab
into
it?
The,
Yeah, this guy.
Sure.
into the oily J-box.
So how long did it take to fix?
It was a couple hours. We had to drain a couple liters of oil out of the J-box, and sorry, I'm pushing buttons here, trying to multitask. And
okay.
so DC vendor is off. Go to hydraulics. That thing have telemetry, does it? Craft power is on.
Coral too.
Should have
That'll work.
telemetry now.
Comes with bonus biology.
I think we call that watch books here. Bonus watch
So
books.
we've been,
I'm floating away.
you got valid comms?
No comms.
So telemetry, so it should right now. I got craft power on.
It just came up.
Okay, so I'm going to turn the craft valve on now. Sorry, we've also been doing this. We go 5, bing, bing. So
Okay.
I always do that most of the time, so I don't have to use the Zeus. And then I was having the Argus pilot hold his finger on the button.
Yeah.
Ooh, it's loose.
It is, yeah. I'm just trying to get a better grip on it. Can you turn on porch light, please?
Roger.
Okay.
Nice one.
Video zoom.
Zoom it in.
It's a big rock.
Okay, I got an index.
Looks good if it'll fit.
Oh, it'll fit.
It'll fit.
Where are we going to put it?
Bonus biology on there. That's going to have to be in one of the big boxes on the starboard side.
They both have box in it already.
Yeah, you could do
You
a
can come
forward box, but I don't know if that's supposed to be reserved for anything else.
What's that?
The forward box Omega.
No,
is,
the front boxes are full of corals.
The
did they
two
put
E and F
another one
and
in
the
there?
starboard bio box are already have big rocks in them.
So this
You can
has a
maybe fit another one.
Omega has a piece of a coral that fell in from another sample, but we don't need it. It's not the only coral that we collected for that sample. But let me review and make sure that that's correct.
I believe the starboard forward is empty, but I believe Steve mentioned, yeah, it's the old geology versus biology thing. I think he wanted to save that for corals.
In starboard?
Okay.
The big
Yeah.
one?
There are
There are
rocks
two. There is a rock in each of the big
in
ones.
In
each
the
of the
forward.
big boxes.
Oh, I said in starboard. Oh, the forward starboard. Sorry,
Yeah, Steve was
miscommunication.
wanting to put biology in the front, and geology in the starboard, but yeah, I'll let you guys work that out. We'll put it wherever you say.
Yes, exactly. Tell me where.
All right.
You might pick up and run while they're waiting to decide, because you're getting
Raj.
run over.
Okay.
All right. I believe Steve said that it's fine, but he's here to confirm.
Yeah,
so,
big man on campus is here.
I can hardly hear
moment.
Taylor in, so just relay what you're. Somebody tell me what she's saying.
Steve just arrived, so he'll tell us if it's okay.
Okay.
All right, confirmed. It's fine to put the rock in forward bio box Omega, so the forward starboard box.
You want to rack back there?
Yeah.
And then before you come in, let me get back to the craft valve page, so if it
Okay.
goes up. And I'll let you get ahead and stop, because that coral might be floaty.
Raj.
Yeah, I don't think we want to be moving while we open the forward one.
Understood.
Might have to come south a bit there.
Yeah.
Say goodbye to your friends.
There you are. Back in the box.
I'm going to sit down and somewhere up here.
I'm going to grab my water real quick.
Yeah, if you just stop, you should be okay.
Okay.
Let her float.
Okay, so rack and back. Center up. Really. Okay. Oops, that's up is down.
I'll let you get the rock a little closer before I open.
Can we zoom out on a high pack now for a minute?
Yep. What did you say, Dan?
Get the rock a little closer before I open.
Okay. I see what you mean. You don't want me to park it?
No,
Okay.
it's C bias is dialed. It'll just sit there.
Okay.
There's not much current.
So like get it
How's
ready
that,
to go
Steve?
in the box?
Yeah.
Yeah, you might tilt
And then
down
open
just a hair for us.
Okay, one more time.
Tilt down.
Get waypoint 4 in there for perspective.
Yeah, that's
Yeah,
good.
okay.
Okay. Are you close?
Yeah, so we're about
Close to where you're-
halfway through the track, something like that. Okay.
Ready?
All set.
Yeah.
Okay.
Oh, wrong button.
You open the starboard, yeah. And just close it when you get a chance. Can you give me it in bubble, please?
It's in bubble.
Oh, I can't. No, it's not very. Like the box.
Oh, you want the drunken bubble?
Yes, please.
Drunken bubble.
You think that's gonna fit?
Yeah, I think it'll fit.
Okay. Me.
You want to come in low and bend your arm up. Otherwise, you'll be in bubble. I come in way a lot lower, like hit the front of the box with it and then lift it up. That way you don't get into the bubble camera. Just like you're doing. Perfect. I don't know.
It looks pretty
Like
big.
the coral recently during dinner.
Oh, it'll fit.
Okay. Come in here. What do you think, Dan? Turn it or? You think that's gonna fit?
I don't think it's gonna fit.
Doesn't appear to like it's gonna fit.
No. We can put it on the porch.
Oh, it'll fall off.
All right, just toss it. It's all right.
Do you want me to try it?
We don't have room in
No,
the
it'll
starboard box?
it'll get stuck.
Okay.
It's
And not on the porch, correct?
not on the porch. There, it's possible to get them back out of the starboard box if they don't fit, but if they don't fit in the forward box, it's really hard to get them back out because it's so tight in there.
Thanks for trying.
No problem.
What's
Next
that?
time, next time.
I had one delivered. What do you want? This one?
No, I was wanting to see my joints a little bit more. But it's
If you
okay.
just lift the shoulder up, now turn it off, it'll fall in.
I know. I like to cradle it in, just in case.
Oh, okay, craft power and valve are off. Bender. Where's that bender? I can never find that darn thing. Okay.
Okay. You
Back,
guys ready to
back
keep
in black.
moving?
Yeah.
Off to the races.
That's gonna be a no sample there for 77X.
Okay. Did you want to try for a different rock in this area or just keep going?
Potentially, if I see one.
Okay. If you see one, yell.
Okay, I will.
Okay.
Maybe don't yell too loud because you can be very, very loud on these microphones, but you know.
I will alert you.
Bridge nav.
Let me get back out ahead
Did
here.
we not want to put it in
Can
the
we move
starboard box?
50 meters
for some
250, please?
I'm not sure, to tell you
Thank
the
you.
truth.
There would be plenty of room. Yeah. Yeah. Don't. Yeah, we can definitely double up on rocks in the starboard EF boxes in the future. Yeah. I think that one would have been too big for the small compartments. It would have been an E or F.
It was.
Oh,
it was bigger
that's all
than
right for now. We'll try and find something more appropriate.
First Victorgorgia of the watch. At least the first one I've seen.
Yeah, they've been seeing them for a bit. They saw them on the last watch a bit.
Oh yeah. First one we've seen, I guess.
Definitely their range.
Do a quick zoom.
Yep, sorry. Just switching over.
We're still moving, right? Moving all the time?
We are moving.
Roger
Yep,
that. Okay.
250.
So, in this purple looking coral,
back up?
is this bamboo coral?
This is its own family of corals. This is a coral called Victorgorgia in the Victorgorgiidae family.
Okay.
What's its most closely related to
Oh, jeez.
I believe
Didn't see that, did you?
bubblegum corals, but I may be wrong about that one.
On a roll tonight.
Trying to think about where it falls out in the clade.
And was that one brittle star around, or was that multiple?
I think that was one. Yep.
Is it more common to see one brittle star or multiple brittle stars around a piece of coral?
It depends on the size of the colony.
Huh.
Altaria?
Altaria.
Yeah. And there's also one over here. But it's from the DVL, which is on the back of the vehicle. So depending on the slope, it's subjective, but I usually use the bubble chem thing there. So we're about 4 degrees, 3.5 degrees pitch down. Maybe a little less on this, depending on how we're horsing it.
Oh, is that its uniform?
Oh, hello there, Jelly. So while we're looking at all these rocks, Brandon, here's a question for you. What is making these rocks look like they're all made? What is making these rocks look like they're made of a bunch of beach pebbles?
That's a good question. So all these rocks are covered in something called ferromanganese, ferromanganese crust, and the morphology of that crust is botryoidal, or you could call it, what would you call it, golf balls?
Just a bit there.